Living world. Can you recall what is the difference between living and non living things? Enlist the characters of living organisms. Whether all organisms are similar? Justify your answer. Planet Earth is made up of abiotic and biotic components. The biotic components are obviously the living being present around us. The question is, why do we call them living and how do they differ from non living? 1.1 Basic Principles of Life. The living being, once produced or born, has to survive. For survival, it needs energy in many chemical molecules. For energy, it has to perform metabolism. Metabolism is breaking of molecules, that is catabolism, and making of new molecules, that is anabolism. From birth onwards, organisms show tendency of growth and development. This growth is a well orchestrated process. You might have observed sand mounds, boulders grow, etc. This growth is not from within, and hence these are not living beings. Growth and development are not the process which have unlimited time span. At certain point of time, the molecules, organs, systems begin to lose their effective working and become old. This is aging process of the body. Life has to continue, hence the organism tries to produce a young one like itself. It is possible due to reproduction, asexual or sexual. This ensures continuity of race. Mules, sterile worker bees do not reproduce yet are living. Can we call reproduction as inclusive characteristic of life? As the body loses its capacity to perform metabolism, the organism dies. Any living being respond to thermal, chemical or biological changes in the surrounding. This is unique property of living being. There is immense diversity in living organisms. Since time immemorial, variety of organisms are living together on earth. In order to understand the interrelation between living and non-living as well as between two living being or groups, systematic study of these is essential. This data is also important for various industries and agriculture. Intensive laboratory and field studies in order to identify and classify the organism form strong basis for meaningful use of collecting data. If we need to study this diversity, certain aids called taxonomical aids can be used. These includes herbaria, botanical gardens, museums, biodiversity parks, etc. Can you recall how can we study large number of organisms at a glance? Whether all organisms prepare their own food? Which feature can be considered as all inclusive characteristic of life and why? Think about it. Can metabolism reactions demonstrated in a test tube called in vitro test be called living? Nowadays, patients are declared brain dead and are on life support. They do not show any sign of self consciousness. Are they living or non living? 1.2 Herbarium. The word herbarium, plural is herbaria, was coined by Piton de Tonfort in the book Elements. The art of herbarium was initiated by an Italian taxonomist Luca Ghini 1490 to 1556. Herbaria are effective tools in taxonomic studies. A herbaria is essentially a dried plant specimen that is pressed, treated, and mounted on standard size sheet in order to preserve it. Date, place of collection, along with detailed classification and highlighting with its ecological peculiarities, characters of the plants are recorded on the same sheet. Local names and name of the collector may be added. This information is given at lower right corner of sheet and it is called label. 1.3 Botanical Gardens. Botanical gardens are the places where plants of different varieties collected from different parts of the world are grown in a scientific and systematic in a in vivo manner. Plants are labeled. The label board shows scientific as well as common name of the plant. Know the scientists. In 1543, first botanical garden of the world was established by an Italian professor Luca Ghini at Pisa, Italy. Botanical garden at Kew in England is known for largest collection of more than 30,000 specimens, preserved plants and more than 7 million herbaria. Conservation of biodiversity. Biodiversity is the degree of variation of life forms in an ecosystem. Biodiversity is essential to maintain ecological stability. The extent of complexity and density of biodiversity can be regarded as a measure of health of an ecosystem. Population explosion and overexploitation of resources has resulted in loss of biodiversity at an alarming rate. Conservation involves attempting to slow down, stop, or even reverse the loss in the natural habitat of organism. This is known as in situ conservation. Why does the loss of biodiversity matter? For many people, it is a simple moral or ethical issue. We share our planet with a huge range of other organisms and we have no right to harm them. Biodiversity helps to maintain stability in an ecosystem. Loss of one variety of organism can affect the entire ecosystem. 1.4 Museum. Museum are the places where collection of preserved plants and animal specimens are kept. Plant and animal specimens may be preserved in formalin, that is 10 to 40% formaldehyde, in transparent jars. Jars are labeled. Larger animals like birds and mammals are usually stuffed and preserved. This science is known as taxidermy. Specimens in dried form are also kept in museums. We can even find systematic collections of shells, skeletons of animals, insect boxes in museums. Thus, biological museums in educational institutes are reference hubs of biodiversity studies. 1.5 Zoological parks. Zoological park, generally known as zoo, is a place of interest for common man. In a zoo, wild animals are kept in captivity. They are protected and cared. They are protected and care is taken to provide conditions similar to their natural habitat. Ex situ conservation. In a zoo, a naturalist can study food habits and behavior of animal. Flora, manuals, monographs, and catalog are some other tools of maintaining biodiversity records. Flora is the plant life occurring in a particular area on time. A monograph describes any one selected biological group, whereas manual provides information keys about identification of species found in a particular area. Can you tell what are the essentials of a good herbarium? Why should we visit botanical gardens, museums, and zoo? What is ex situ and in situ conservation? 1.6 Biodiversity parks. It is an ecological assemblage of species that form self sustaining communities on degraded or barren landscape. Example Late Utamrao Patil Biodiversity Park, Gureghar, Mahabaleshwar. This park is the best model for conservation of natural heritage in urban landscape. Systematic classification of living organisms is helpful in understanding the interrelation. In order to understand the interrelation between organisms and maintain harmony on planet Earth, study of biodiversity is a must. Know the scientist. Dr. S.P. Agarkar. One of the leading botanists of India, Dr. S.P. Agarkar was born in November 1884 in Malvan, Maharashtra. He explored biodiversity of Western Ghats where he came across a species of freshwater jellyfish which was until then only known to be found in Africa. These findings were published in scientific journal Nature in 1912. Dr. Anadale, the superintendent of the Indian Museum in Kolkata, helped Dr. Agarkar in his further endeavor to collect, preserve, and conduct microscopic examination of animals and plant specimens. The Institute ARI Pune has been named after his name. 1.7 Key. Key is taxonomical aid used for classification of plants and animals. The key are based on contrasting characters. One of the contrasting characters get accepted and other rejected. The statement in key is called a lead. Normally keys are analytical in nature. Let us study about classification of living organism in next chapter. Do you know when plants from any forest locality are conserved on the name of holy place, it is called as sacred groves. These are also considered as sacred natural sites by IUCN. You can find out these things that is given. This is the exercise that you can solve. Thank you.